including membrane inhibitor of reactive lysis MIRL or CD59, also acts to block formation of the membrane attack complex. MIRL is widely distributed on the cell membranes of all circulating blood cells, including red blood cells, and on endothelial, epithelial, and many other types of cells. Its main function is to bind to C8 and prevent insertion of C9 into host cell membranes. The table 6-3 lists the receptor and indicates the type of cell on which they are found. Now let's talk about the complement receptors and their biological roles. Some complement receptors found on host cell amplifies and enhance the immune response by augmenting phagocytosis and stimulating successory cells, rather than acting as regulators. Table 6-3 summarizes the main characteristics of each and describes the cell types on which they are found. CR1 has been discussed in the previous sections. A second receptor, CR2 or CD21, is found mainly on B lymphocytes and follicular dendritic cells. Ligands for CR2 includes degradation products of C3b such as C3DG, C3D, and IC3b. In addition, the Epstein-Barr virus gains entry to B cells by binding to this receptor. CR2 is present only on mature B cells and is lost when conversion to plasma cell occurs. Now, these are the C receptor on cell membrane for complement components. We have CR1, or known as the CD35, has a ligands of C3b, IC3b, and C4b. These cell types are RBCs, neutrophil, monocytes, macrophage, eosinophil, B and T cells, follicular dendritic cells, which their function is the cofactor for factor 1 mediates transport of immune complexes. CR2, on the other hand, also known as CD21, has a ligands of C3DG, C3D, IC3b. B cells, follicular dendritic cells, epithelial cells are their cell types. Its function is that B cell co-receptor for antigen with CD19. Now we have the CR3, also known as the CD11B and CD18, which it has a ligand of IC3B, C3D, and C3b, which their cell types is monocytes, macrophage, neutrophil, and natural killer cells, which its function is the adhesion and increased activity of phagocytic cells. CR4, or the receptor also known as the CD11C or CD18, has a ligands of IC3B and C3b, which its cell types is the monocytes, macrophage, neutrophil, natural killer cells, activated T and B cells, and dendritic cells, which its function is the adhesion and increased activity of phagocytic cells. The DAF or CD55 has a ligands of C3b and C4b, which its cell types is RBCs, neutrophil, platelets, monocytes, endothelial cells, fibroblasts, T cells, B cells, epithelial cells, which its function is to dissociate C2b or BB from the binding sites, thus preventing formation of C3 convertase. MIRL or CD59 has a ligands of C8 only, and the cell type or the RBCs, neutrophil, platelet, monocyte, endothelial cell, and epithelial cells, which it prevents insertion of C9 into cell membrane. And the last but not the least is the MCP or CD46, also known as the CD46, which it has a ligands of the C3b, C4b, which it has cell types of the neutrophil, monocytes, macrophages, platelets, T cells, B cells, and endothelial cells, which its function is to cofactor for factor 1 cleavage of C3b and C4b. Now, let's talk about CR2. CR2 plays an important role as part of the B cell co-receptor for antigen. Acting in concert with CD19, it binds complement-coated antigen and cross-link it to membrane immunoglobulin to activate B cells. In this manner, immune complexes are more effective at enhancing B cell differentiation and production of memory cells than is antigen by itself. Another receptor, CR3 or CD11B, CD18, found on monocytes, macrophage, neutrophil, and natural killer cells, specifically binds particle opsonized with IC3B, a C3P degradation product. It does this in a calcium-dependent manner. The CR3 receptor plays a key role in mediating phagocytosis of particles coated with these complement fragments. It consists of an alpha and a beta chain with a molecular weight of 165,000 and 95,000 respectively. This protein triggers surface adhesion and increased activity of phagocytic cells. Patients whose white blood cells lack this receptor fails to exhibit functions such as chemotaxis, surface adherence, and aggregation. Deficiencies in phagocytosis are also noted. This individual have an impaired capacity to bind IC3B coated particles and are subject to recurrent infection. The CR4 or CD11C/CD18 receptor is very similar to CR3 in that it also binds IC3B fragments in a calcium-dependent fashion. CR4 proteins are found on neutrophil, monocytes, tissue macrophage, activated T cells, dendritic cells, natural killer cells, and activated B cells. Neutrophil and monocytes, however, possess smaller amounts of CR4 than of CR3. Their functions appear to be similar to that of CR3, and they may consist neutrophil adhesions to the endothelium during inflammation. Receptors specific for CLQ are found on neutrophil, monocyte, macrophages, B cells, platelets, and endothelial cells. This receptor, known as colectin receptors, binds the collagen portion of C1q and generally enhances the binding of C1q to Fc receptor. Interacting only with bound C1q, the receptor appears to increase phagocytic cells uptake of immune complexes opsonized with C1q. Additionally, on neutrophil, they may act to enhance the respiratory burst triggered by IgG binding to Fc receptor. Now let's talk about the biological manifestation of complement activation. Activation of complement is a very effective means of amplifying the inflammatory response to destroy and clear foreign antigens. The cycle does not always have to be proceed to lysis for this to be accomplished. Hence, some of the initiating protein are much more plentiful than proteins that forms from the membrane attack complex. Complement protein also serve as a means of linkage innate and natural immunity. They act as an opsonin to facilitate destruction by phagocytic cells, and they play a major role in uptake and presentation of antigens so a specific immune response can occur. They also facilitate B cells activation and may in fact be necessary for maintaining immunological memory. Effectors molecule generates earlier in the cascade played a major role in all these areas. Such molecules can be classified into three main categories: anaphylactoxin, chemotaxins, and opsonin. An anaphylatoxin is a small peptide that causes increased vascular permeability, contraction of smooth muscle, and release of histamine from basophils and mast cells. Proteins that play such a part are C3A, C4A, and C5A. All of these All of these have a molecular weight between 9,000 and 11,000, and they are formed as cleavage product from larger complement components. Of these molecules, C5a is the most potent, at least 200 times more powerful than C3a, while C4a is the least effective. C3a, C4a, and C5a attach to specific receptor on neutrophils, basophils, mast cells, eosinophils, smooth muscle cells, and vascular endothelium. C3a and C4a attach to the C3a receptors or C3AR, and C5a attaches to the C5a receptors or C5AR. When binding occurs on basophil and mast cells, histamine is released, thereby increasingly vascular permeability and causing concentration of smooth muscles. C5a causes neutrophil to release hydrolytic enzyme, oxygen, radicals, and prostaglandins, aiding in destruction of foreign antigens. C5a also serves as a chemotaxis for neutrophil, basophil, eosinophil, mast cells, monocytes, and dendritic cells. In this manner, these cells are directed to the source of antigen concentration. Because of increased vascular permeability, neutrophils migrate from blood vessels to the tissues and tend to aggregate. Binding of C5a to monocytes cause them to undergo an oxidative burst that includes increased production of hydrolytic enzymes, neutrophil chemotactic factor, platelet activating factor, interleukin-1, and toxin oxygen metabolites. Interleukin-1 is a protein that enhances T cell activation. This may produce fever and an increase in acute phase reactant, both of which are characteristics of an inflammatory response. As a means of localizing and controlling the effects of C3a, C4a, and C5a, these substances are rapidly inactivated by an enzyme in the plasma called carboxypeptidase. And C3a and C4a are cleaved in seconds. While conversion of C5a occurs more slowly, the last major effects of complement-derived peptides is opsonization. C4b, C3b, and IC3b, which accumulates on cell membrane and as complement activation proceeds, binds to specific receptors on erythrocytes, neutrophils, monocytes, and macrophages as discussed earlier. This facilitates phagocytosis and clearance of foreign substance, and it is one of the key functions of the complement system. In addition, attachment of C3 products to an antigen has been found to enhance the B cell response. Complement. Now we're going to talk about the complement in disease states. Although complement act as a powerful weapon to combat infection by amplifying phagocytosis, in some cases it can actually contribute to tissue damage or death. Complement can be harmful if activated systematically on a large scale, as in gram-negative septicemia. It is activated by tissue necrosis such as myocardial infarction or lysis of red cells occurs. In the case of septicemia due to a gram-negative organism, large quantities of C3a and C5a are generated, leading to neutrophil aggregation and clotting. Damage to the tiny pulmonary capillaries and interstitial pulmonary edema may result. Tissue injury following obstruction of the blood supply, such as occur in myocardial infarction or heart attack, can cause complement activation and deposition of membrane attack complex on cell surface. Receptors for C3a and C5a have been found in coronary plex, indicating the complement components may increase the damage to heart tissues. Lysis may be another end result of complement activation. Hemolytic disease such as cold autoimmune hemolytic anemia are characterized by the presence of an autoantibody.